Hello everyone, welcome back to another class on regeneration biology. In today's class, we will learn about the basics of regeneration put into practice in vitro. So what are the developments we have made? What are the pros and cons which we end up getting while addressing the regeneration when you try to put in place in a laboratory condition, that is in vitro. So we know we have lots of animal models of regeneration. We have been practicing or understanding their regenerative approach extensively, and it is a very fascinating biological phenomenon that continues to intrigue a human being or humanity for centuries. We know many researchers have started working with not you cannot call it as a research. You decapitate the snail's head and the head will grow back, or cut the planaria into pieces, the lost head will come, and those kind of research people have been intrigued by. Lots of researchers or layman, and people have also observed the lizard's tail is growing, etc. But no one could comprehend even today that the exact depth of it. If you understood each and every step of it, then you would be able to simulate the same thing in human or any other mammal. We haven't reached up to that stage, although the knowledge is piling and building. The study of regeneration promises to inform how adult tissue heal and rebuild themselves, so that this process may someday be stimulated in a clinical setting. That is the overall idea, overall goal of studying this so-called regeneration biology. Although mammals are limited in their ability to regenerate, closely and distantly related species can perform an astonishing regenerative feats. Many examples are there, although mammals cannot. Some examples like spiny mice can regenerate their skin naturally, and also lots of regeneration studies are undertaken in that model as well. So many animals representing almost all phyla harness an innate ability to rebuild the missing adult structures lost because of an injury. And this replacing the missing structure can be perfect replacement or running the show, like stopgap arrangement. Both ways you can see it. However, which regeneration aspects are conserved and unique to a given context is unclear. For example, if you think zebrafish can regenerate, but Japanese Madaka cannot regenerate, which is also a bony fish, which is also a aquatic animal. So we cannot assume that if it is a fish, it can regenerate. Same way, mammal. Mus musculus cannot, but Acomis species spiny mice can regenerate to a reasonable extent. So it varies from individual to individual, and all individual, what I mean is individual species. In all other scenario, every organism can regenerate its liver, and it can regenerate its RBC, because blood cells, WBC, they are all limited in their capacity. Their lifespan is limited. So if you cannot regenerate your blood, you are dead. Or if you cannot regenerate your damaged liver, you are dead. So every organism have got a reasonable regenerative capacity, but it varies. Not that every organism have got 120 days of lifespan for their RBC. In humans, it is 120 days, whereas other animals, it varies. So one aspect of regeneration that appears to be shared is using stem and progenitor cells to replace the missing tissues. No animal can directly make a differentiated tissue from a pre-existing differentiated tissue. Say one RBC cannot be formed from another RBC. You need to have its hematopoietic stem cell or the so-called progenitor cell. While many animals regenerate, we limit our discussion to mainly to amphibians, zebrafish, planarians, because they are well-studied bilateral organisms, or well-studied, which shows the bilateral symmetry, and they invoke the so-called cell proliferation in response to injury. Say Hydra has got a morphollaxis which will rearrange the cells and form a miniature hydra, and then they give rise to, it will keep growing by cell proliferation. Whereas other animals, they form the so-called blastema, which is formed by cell proliferation, and it gives rise to the damaged tissue. Except for planarians, the cellular source of regeneration remains mysterious. Mysterious in the sense it regenerates, we know that it forms from the reprogramming of the existing cell. But there is also possible, like we discussed about how stem cell from other tissue is moving on to a different tissue and then fixing it. So these are all possibilities. But in a well-regenerating organism such as zebrafish, the differentiated tissue, say heart tissue, cardiomyocytes reprogram and give rise to stem cell. In retina, the Mueller glia reprogram and give rise to the stem cell. So in planaria, each tissue present in its limb or tail is reprogramming and give rise to each types of cells. So we know that. But planarians have got a dedicated, around 30% of planarian body have got this cineoblast, and which is capable of regenerating constantly. And the reason also we discussed, because every organ in planaria is short-lived, somewhat like that of our blood. Our blood is constantly revamped because none of the blood cell present in your body is more than 120. The oldest blood in your body is 120 days old, which will die today evening or tomorrow. And it will start forming new cells soon. So we should understand none of the organs present in planaria are old. It keeps revamping every now and then. Are stem cells that rebuild missing tissues present before the injury, or are they generated during the injury response? One question one can have. If they are generated de novo, where do they come from? Are they coming from the site of injury, or they are coming from elsewhere? Another question one can ask. Can regenerative stem cell give rise to all missing cell types, or multiple lineage restricted stem cells are required? This is another question we can have. And the current studies in animal regeneration are intensely focused on answering these central queries. Some animals we know, like zebrafish we know, axolotl we know that where from these stem cells are forming. But in other animals where we know some regenerative ability, we always wonder where are these stem cells coming from. Let us see a few facts. For an adult animal to replace the missing structures with an exact copy of what is missing, it is clear that the developmental programs must be redeployed, because an organ is formed during development. So if the same organ that is to be formed, we must understand it should rekindle the regenerative response only via the developmental program or the embryonic approach. However, the dynamics of cell communication and proliferation are vastly different, and as the cell types are involved, depending upon which tissue, which organ you are making, accordingly it will change. To accomplish regeneration, adult animals may invoke the proliferation of differentiated cells, the activation of reserve stem cell, the formation of new stem cells with limited capacity to self-renew, that is the cell progenitor cells, or a combination of these strategies. Depending upon the species, depending upon the organ, you can apply any of these approaches. Let us see a few more facts. Which cells in an adult animal divide and differentiate to replace multiple cell types required during regenerative response? So what is that starting cell type? While this is basic fundamental question formulated and reformulated through successive generations of biologists, its resiliency against experimental attacks have proved surprising and in many cases quite frustrating. Although we see something regeneration, but we are not able to come up with a concrete set of events that trigger this whole process. Nonetheless, we should understand that different tissues, both within the same organism and the same tissues of the different organisms, use different strategies to achieve tissue repair or regeneration. Not that there is a Swiss Army knife kind of approach. There is no standard operating procedure for every tissue. It varies from tissue to tissue, organ to organ. For example, the vertebrate liver invokes compensatory regeneration after removing 2 liver lobes, whereby the remaining lobe proliferates to acquire the original tissue mass without replacing the missing lobes. So it is called a compensatory regeneration. So regeneration can be compensatory in the case of liver, tissue specific in the case of heart and skeletal muscle, liver, pancreas, lens, retina, etc. Or it can rebuild complex structures containing multiple tissue types or organ types such as limbs, fins, tails, etc. So species vary, organ vary. Accordingly, it can have a different way of approach for implementing the overall regenerative response. Now let us quickly see the dedifferentiation. So the term dedifferentiation is often employed the regeneration literature to describe the loss of differentiated characters of cells after amputation or an injury and their concurrent acquisition of an undifferentiated morphology during blastema formation. So in nutshell, an existing cell is going back in time, means it is going few steps behind, so that it will be able to now lose the existing differentiated character. So it gets dedifferentiated, so that it will be not necessarily come back through the same route and give rise to this cell type from which the dedifferentiated cell originated, but it can give rise to another cell type. So the dedifferentiation process could be the release of living cells from the confines of their previous organization with accompanying active mitosis of these cells. In a simple way, you can define that way. It is moving away, in a different direction from this existing differentiated identity. However, the term dedifferentiation is easily misinterpreted to imply that a cell has attained a multipotent undifferentiated state. That is not always correct, because there is currently insufficient evidence to either suggest or rule out a major role for reserving stem or progenitor cells in urodel blastema formation. The term dedifferentiation, as originally coined, may therefore refer to a reversal of the differentiated state, just a reversal. Sometimes it will still have the differentiated cell specific proteins, but the level may be low in the dedifferentiated state. An activation of reserve stem cell or progenitor cell status, or a combination of both. It has a differentiated cell do not have any progenitor gene turned on. It has lot of tissue specific genes turned on. So the tissue specific genes will be turned down, not off, turned down, and it will turn on lot of progenitor specific genes, and both these genes may stay together, and eventually the progenitor specific genes may pick up and the differentiated cells may completely shut off. Then one can claim, oh, this progenitor became completely moved away from the differentiated status. What is the differentiation potential of cells present in the blastema? Let us quickly see through this picture. The blastema may be composed of 1. Cells that are restricted to give rise to the same tissues from which they were derived. 2. The cells that were multipotent and give rise to different tissue types. And 3. A complex mix of cells with a variety of origins and potentials. These are all the possibilities. In a larval tail of the urodel, say let it be salamander or axolotl, anything, fluorescently tagged glial cells of the spinal cord, they proliferate during regeneration and they can give rise to tissue outside the spinal cord. Originated from spinal cord, it has a tag. Now it is not giving rise to just a spinal cord, it can give rise to other. Data currently indicates that at least in larval tissues, the urodel blastema contains a complex mix of lineage restricted and multipotent cells. They are present. However, very little is known about the potential of blastema cells in adult urodels. In anurine amphibians, some example we have studied in the bone marked with the GFP etc. in the previous lectures. You can revisit that. GFP positive grafts into GFP negative host illustrate that the cells of the regenerating spinal cord and notochord are derived from cells of those very same tissue in adult. In bone also it will give rise to only bone derived stem cells give rise to the new bone. It doesn't give rise to other cell types. But in larvae it can be different. Larvae it can give rise to a mix of other tissues also. Spinal cord derived cell can give rise to other cell types also. Therefore, the progenitor cells during anurine tadpole regeneration appear to be restricted in their potential. Like you can see here, blastema cells, once they mark it, like in this picture you can see, in once you mark them, they specifically give rise to that particular cell type from where they have originated. But in other, this B and C we can see in the next panel. So B is a multinucleated myofiber. How myofiber react? In panel B, injection of dye into multinucleated muscle fibers prior to amputation illustrates that once clipped or once injured, some of these fibers can fragment into proliferating mononuclear cells that contribute to the blastema formation. The long-term fate of these cells is still under active investigation. We can see here they have clipped it and mononucleated cells because it is already tagged, and you can see how they are migrating. And in C, the muscle fibers in adult newts contain satellite-like cells that express PAX7 genes, which is seen in green in color. And they are separated from the rest of the cell by the basement membrane, which is shown red in color. And middle right here you can see both are present. When these cells are isolated, cultured, they are already tagged in red color and implanted into regenerating newt limbs, they contribute to the blastema and they give rise to unrelated tissue types including cartilage and epidermis. This is also one has to keep in mind. In adult, although it is restricted, or in a given tissue it can be restricted, in some other tissue they can be moving to other lineage also, some kind of transdifferentiation. Now let us see in vitro studies. Do we have some examples? Muscle cell culture experiments focus on newt A1 and mouse C2C12 cells. Okay, two different cell lines. A1 cells from the newt and C2C12 from the mouse. Both mononucleated cells, can be induced to fuse and form myotubes. Upon differentiation, they are individual cells. They will fuse and form a myotubes if you give the right cue. In culture, upon serum deprivation. One approach is serum deprivation. They will form muscle fibers. These experiments uncovered a key difference between the newt and mammalian in vitro derived myotubes. Both are myotubes, both are cells. Although names are different, both are biologically they are muscle precursors. What is the difference? Newt A1 myotubes will enter the S phase when exposed due to high levels of serum. S phase means DNA replication, or lower levels of thrombin treated serum. A1, that is from the newt or from the frog or from the salamander. At the same time, the mouse, which is a mammalian, C2C12 cells do not respond, indicating the presence of an active signal in animal serum that leads to proliferation of A1 myotube nuclei. S phase means proliferation, and whereas C12 cells doesn't respond, whereas newt can respond to something what is present in the serum and it enters the proliferation. However, let us note further. It should be noted that the extent of differentiation and therefore the relative developmental starting point for the two cell types remains unclear. What made them different? Although functionally they are becoming myotubes eventually. For example, newt A1 cells or these precursor cells of the myotubes do not exhibit striation or peripheral alignment of the nucleus, like when the syncytial nucleus is formed in a myofiber, myotubes. In addition, in vivo urodil amphibian muscles contain satellite-like cells that express PAX7, which is a marker, and are separated from the myofiber by a basement membrane. So this is what you see in vitro and in vivo variation. Let us see further. Forced expression of another gene, MSX1, a homeodomain protein which is known repressor, which has a repressor function in C2C12, that is a mouse cell. C2C12 cells, myotubes, causes a small fraction, around 5% of the cell, to fragment into proliferating mononuclear cells. This has been seen. Under proper culture conditions, these cells can differentiate into other cell types such as adipocytes, chondrocytes, myocytes, and osteocytes. They give rise to different like fat, cartilage, muscle, and bone tissues. Because the C2C12 cells are multipotent, these results should be treated with a little bit of caution. Nonetheless, this was a key demonstration that mammalian myofibers can be induced to reverse their differentiated state. That is what the whole point is about. Complementary studies were also carried out in newts in which the primary larval limb myofibers are normally fragment upon dissociation where they are inhibited from doing so via MSX knockdown. If you don't allow MSX to be there, then the blastema formation will be jeopardized. Given the caveats mentioned above for cultured cells, the data argue that MSX1 may be necessary and sufficient for differentiated muscle to fragment into proliferating mononuclear cells, which is a necessity for the blastema formation. To the contrary, in vivo morpholino-mediated knockdown of MSX1 in individual larval axolotl tail muscle cells had no negative effect on the ability of these animals to fragment. This is showing an opposite result. That means even if MSX is missing, some other genes was able to contribute to this fragmentation process. This discrepancy has several potential explanations, including but not limited to the differences likely exist between in vitro and in vivo conditions, naturally, because in vitro petri dish condition is different from the live animal condition, and that muscle cells of the limb and tail could exhibit a differential requirement for MSX1 expression during fragmentation. During fragmentation, both whichever, depending upon which species and which tissue you are talking about, MSX dependency can vary. Let us see a cardiac example. In implantation experiments, cells isolated from the newt heart, cardiomyocytes, and in short form they are called CMs, were tagged and implanted into unamputated or amputated limbs. Took cells from heart and put it into the limbs. While the cardiomyocytes implanted into unamputated limbs were stable and exhibited no special behavior, they were activated when implanted into day 5 regenerating limb blastomas, and 65% gave rise to skeletal myotubes. Means from heart it became skeletal muscles. At the same time, if you expressed a cartilage cell marker, this is clear evidence of the plasticity of the differentiated status in the adult urodil amphibians. Still, the magnitude of fate change is unclear because of the transplanted cardiomyocytes continue to express desmin, which is a marker found in many muscle cell types. So while these experiments do not rule out the role of reserve stem cell progenitor cells, they clearly illustrate that a large fraction of isolated newt cardiomyocytes can at least switch muscle cell types. Heart to skeletal muscle may sound little, but it is very enormous when you think about a cell type. Proliferating cells derived from satellite-like precursors were isolated in culture from adult newt myofiber explants. This is another set of experiment done in salamanders. When tagged and implanted into regenerating adult limbs, these cells contributed to the blastema, and many appeared to switch lineage into cartilage and even epidermal cells, the lineage switching. On the contrary, another group of researchers, they found that implanted primary myofibers from juvenile axolotls can fragment and proliferate without even the satellite cells. They are not necessary for them to proliferate. Implanted primary myofibers from the juvenile axolotls can fragment and proliferate even without any other inclusion of the satellite cells. It is possible that these disparate or the variation in results can be explained by the difference in the species and the life cycle stage, whether they are juvenile versus adult, or by the different criteria used to assess whether the satellite cells were present or not. That is the question that was asked. It is also possible, remains to be observed, that it is possible that both myofiber fragmentation and satellite cell proliferation contribute to the blastema in vivo. In reality, that is what is observed. And their relative contribution may be age and species or tissue specific manner, it can vary. Now, if you see an overview, in vitro animal regeneration involves studying and replicating regeneration processes outside a living organism, typically in a lab setting. That is why in vitro studies has to be monitored and validated in reality in a in vivo condition. This approach allows researchers to isolate specific factors and mechanisms involved in regeneration and manipulate them under controlled conditions. The basics of animal regeneration, such as stem cell activation, dedifferentiation, and transdifferentiation, can be explored in vitro very effectively than in vivo using techniques like cell culture, organoid models, and tissue engineering. That is why the in vitro approach becomes very fascinating. To elaborate further, stem cell activation and differentiation, many animals utilize stem cells to regenerate damaged tissues. In vitro, researchers can study how these stem cells are activated, proliferate, and differentiate into new cell types needed for regeneration. For example, they might investigate how certain growth factors or signaling molecules promote stem cell activation and differentiation in a dish so that can be put into use in an organ culture. Dedifferentiation and transdifferentiation. Some animal species can regenerate by inducing cells to lose their specialized characteristics, such as this process is dedifferentiation, and then differentiate into a new cell type, that is via transdifferentiation. In vitro, researchers can examine the factors that trigger these processes, such as changes in gene expression, exposure to specific chemicals, etc. That is the beauty of in vitro study. Tissue and organ regeneration. In vitro, researchers can also attempt to regenerate entire tissues or even small organs. This involves creating a 3D environment like an organoid that mimics tissue structure and providing the necessary signals for cell proliferation and differentiation. For example, researchers might use some cells to build up the components that is such as a small intestine or lung, etc., or a muscle fiber. And in vitro models and techniques, when you use various in vitro techniques are used to study and replicate the regeneration what occurs in the in vivo scenario. This includes cell culture, that is growing cell from an animal species in a dish to study their behavior and potential for regeneration, and organoid models, that is creating 3D structures that mimic the architecture and the function of an organ and that allow more complex regeneration studies. And then comes the tissue engineering itself, that is using scaffolds and the cells to create new tissues or organ in a lab setting. So if you look further, the applications include in vitro regeneration research has a tremendous potential to lead to new regenerative medicines and therapies, because organoid culture and the scaffold and the stem cell on scaffold, etc., is nothing but in vitro system. By understanding the mechanisms of regeneration in vitro, scientists can potentially develop treatments for injuries, diseases, and conditions that the tissue regeneration is limited or absent. So the idea is, when you look into a in vitro system, you are trying to learn about how a given tissue is interacting, or a given cell type need help from another cell or another cytokine. Can we increase or decrease? These are all very much possible in an in vitro study. This should be taken into a real scenario for in vivo help to a patient or a organism model which you are studying. So we will learn more in detail about the different aspects of regeneration in the next class. Thank you.